This video we are going to talk about the messenger RNA processing and use. The first is the alternate splicing. In the alternate splicing, we alternately splice exons of different kinds. And I've already explained that in a previous video, so I'm not going to go into details. The example is of tropomyosins. Then we have alternate polyadenylation. I'm going to go over it briefly. In alternative polyadenylation, for example, this is our mRNA. Our mRNA is going to have different poly A tails at different sites. And it is now going to get alternately cleaved, and that is how you will have different mRNA transcripts from one mRNA. Then we have the mRNA editing. In mRNA editing, we have is that the mRNA, after being fully transcribed and fully, there are all the modifications are done, and still mRNA can be altered. And this is when we have two types of in VLDLs we have two types of ApoB proteins. We have ApoB100 and we have ApoB48. So this is in the liver and this is in the intestine. So what happens in the intestine is that the bigger, longer mRNA, it is cleaved by, when it is in the intestine, the cytosine, it is replaced by a uracil, and cytosine was supposed to code for glutamine. It was supposed to code for glutamine, but it is deleted and replaced by uracil, and now this will give you a stop codon, and that is how you will have a truncated or a smaller protein, that is the ApoB48. That's mRNA editing. And the next is the mRNA stability. Now in this example, we have the first one is the iron metabolism. In the iron metabolism, we have two proteins that we're going to talk about. One is transferrin and the other is ferritin. So transferrin, I will first tell you a bit about transferrin. So transferrin, it transports or carries iron in plasma. So it is in the blood, and ferritin is stores iron in cells. Okay, now first we're going to talk about transferrin. So what does transferrin do? The transferrin attaches itself to a receptor called as the TFR, or transferrin receptor. We call it TFR. Now we're going to look at the expression of this receptor, not transferrin receptor, the transferrin receptor. Okay. So in the transferrin receptor is a protein. Okay. So it's mRNA, not DNA. It's mRNA. We have its, I'm going to draw its mRNA. We have its coding region and its untranslated 3' end. So this is the untranslated 3' end. It has iron response elements. So these loop-like structures are iron response elements. Remember, these response elements are in the mRNA, not like before we said that they were in the DNA. They are in the mRNA. And these iron response elements, they are gonna get bound by a regulatory protein called as the iron regulatory protein. These are not special transcription factors, you guys. These are just regulatory proteins that are going to bind to the IRE. So when they are bound, this is going to increase the transcription. It is going to increase the transcription of the TFR. So when the cells, I'm going to do that in a different color, when cells have low iron, the iron regulatory proteins are going to increase, and they are going to bind to the iron response elements, and they are going to increase transcription, and TFR will be made so that the iron can be transported into the cells. But when cells, I'm going to do that in orange, iron is high, then it is going to down regulate the iron regulatory proteins, and these are not going to bind into the iron response elements, and transcription will be less, and TFR made will be less, right? And now the same story for ferritin. For ferritin, now ferritin, it is a protein inside the cell, so we are talking about the expression of ferritin, whether ferritin will be increased or decreased, okay? Not the receptor, it's the ferritin itself. Now for ferritin, when the cell's iron is low, we don't need ferritin, right? Why do we need ferritin if we don't have iron to store, right? So what they're going to do is that it is going to decrease, cell iron is low, they're going to decrease the iron regulatory proteins, and it is going not bind to iron response elements, and there is going to be decreased ferritin, and vice versa. Okay, the next example we have the RNA interference. Now RNA interference is basically that the RNA can be, RNA is the mechanism of gene silencing. It is the mechanism of gene silencing. How do we do that? We can do in two ways. Either we can decrease transcription, or we can destroy mRNA, right? Now how do we do that? What happens, I'm gonna show you how to destroy RNA, okay? Not how to decrease transcription. We're gonna do how to destroy RNA. This is our DNA. Let's make it DNA like. This is our DNA. It is going to make our, let's say, primary RNA, right? So this is our primary mRNA, or you could say micro RNA, right? There is an enzyme called as DROSHA. So this is DROSHA enzyme. This is going to convert the primary microRNA into pre-microRNA. This occurs inside the nucleus. Then the pre-microRNA is going to travel into the cytosol, and here we are going to give it dicer. It is another enzyme, and this dicer is going to convert into microRNA. Now this microRNA, this is a double-stranded microRNA, that is important. So this is a double-stranded microRNA. The primary microRNA is like a loop structure, and pre-microRNA is like a loop structure also. But, excuse me, I'm very sorry. But the microRNA is a double-stranded structure, not a loop structure, okay? And now this microRNA, this is our main guy. This is going to go on and destroy target mRNA. That is that simple. It is going to either degrade the target RNA, and those enzymes by which it could do is argonoate or AGO, or we can call it slicer. So it degrades or destroys the target RNA, or it could just repress the translation. You can do one of the both things. There are certain therapeutic things that we can also do for RNA interference, and it is a disease called as the age-related macular degeneration. And age-related macular degeneration, what actually happens is that there is overproduction of VEGF, which increases blood vessels, and increase in blood vessels causes blindness. So we can use RNA interference to decrease this VEGF, and and blood vessels will decrease, and it will prevent blindness. But it's still in the clinical trials, as the book suggests, and it has not yet made for medical use.